All right, so this picture is a little bit blurry, but I'm just going to do a brief overview of meiosis and go over a little bit of interphase because I don't really go over that in my next recordings, and I just want to clear some stuff up just so it's pretty clear. So in meiosis, we're going to look at the right-hand side of this picture, and in meiosis, what happens at first is that we are going to have interphase occur first. And in interphase, we have DNA replication and growth and development. This is where that occurs. And then after interphase and DNA replication occurs right here, we are going to go into meiosis 1. Now, in meiosis 1, we have prophase 1 through cytokinesis at 1. So in all of those steps, we're going to have division, and it's important to note that we're going to have division of homologous chromosomes. So let's say that we start off, here's the original cell. We'll say n equals 4. Then after DNA replication occurs, we have a cell with DNA that's replicated. So it's going to be 2n equals 4. There are still 4 chromosomes, but they're simply just replicated, and they're going to look like these X-shaped things. Usually that's how they're depicted when they're condensed. So we have 4 chromosomes that are replicated going into meiosis 1. Meiosis 1. And then after meiosis 1 occurs and we have that first round of division right here, we're going to get 2 haploid daughter cells. And these cells are going to be genetically different because, we had independent assortment and crossing over it in meiosis 1 during prophase and metaphase. And so we're going to have 2 new daughter cells that and n will equal 4. So we have these daughter cells that now have unreplicated DNA, and they're haploid because we've split them apart, split the homologous chromosomes apart, but they technically have the same amount of chromosomes as that original daughter cell. And that is completely intentional that way because we don't repeat replication as we go into meiosis 2. We're going to result in 4 cells that have half the DNA of the parent cell. So that leads me into our next part, which is meiosis 2, which it consists of prophase through cytokinesis 2. And so DNA is not replicated again. So, DNA is not replicated again. And so this results in us getting 4 of these unique haploid daughter cells. 1, 2, 3, 4. And they're unique, and they have the half the number of chromosomes that the original cell does, and that is n equals 2. So I just wanted to do a quick overview of that just because I talk really fast in my video, but I wanted to get the understanding that interphase occurs with replication only occurs once, which is why meiosis and having these gametes is possible. In prophase 1, the replicated DNA condenses into X-shaped structures, and these structures are called homologous chromosomes, and they pair up. And when they do pair, this is when crossing over or recombination occurs, when the parts of the chromosomes swap parts. By the end of prophase 1, the nucleus is disappearing and the spindles are extending across the cell. In metaphase 1, the pairs of homologous chromosomes line up at the equator of the cell and the spindles attach to the homologous chromosomes. Now it's important to note that the spindles are not attaching to the centromere that connects the sister chromatid. They are attaching to the homologous chromosome. In anaphase 1, the homologous chromosomes are pulled to opposite poles of the cell. Then in telophase and cytokinesis 1, which often occur at the same time, the nucleus reforms and the cell then is pinched into two at the cleavage furrow. Now the chromosomes do decondense sometimes, but often this step is skipped because another round of division is about to occur. The end of meiosis 1 results in two newly formed haploid daughter cells, each with 4 chromosomes. At the end of meiosis 1, the DNA does not replicate. In prophase 2, the nuclear membranes in both cells break down and the chromosomes condense, and the spindles are spreading across the cell. In metaphase 2, the chromosomes individually line up at the equator, not in pairs, and the spindles attach to the centromeres. Anaphase 2, the sister chromatids are pulled apart to opposite ends of the pole. In telophase and cytokinesis 2, the nucleus reforms and both cells split into two at the cleavage furrow. This results in 4 haploid daughter cells, each with half the DNA of the original parent cell, where n equals 2.